You are listening to Medicine and the Machine with Medscape Editor-in-Chief Eric Topol and Master Storyteller and Clinician Abraham Verghese as they talk with experts around the globe about the hottest topics in healthcare. This podcast is intended for U.S. healthcare professionals only.
Hello, it's Eric Topol with Medicine and the Machine, our Medscape podcast, and I'm really delighted today to have Dr. David Fajgenbaum join me from the University of Pennsylvania, Penn Med, and has a remarkable story that will be partly familiar to some of you in the medical community, but there's a lot more chapters going forward. So welcome, David.
Eric, thanks so much for having me.
Well, let me just start off with, I recently had a chance to read your amazing book, Chasing My Cure. I mean, it is quite a story, and I guess maybe, well, we go back to 2010 when you picked up that something wasn't right about your health, even having been like the picture of health, bench pressing 375 pounds, the beast. But then something went off the track, and maybe we could go start at that point.
Sure. So I was a healthy third-year medical student, just like you mentioned, never had any problems in my whole life. And then out of nowhere, I just started feeling more tired than ever before. And you remember during your training, we're sort of always all tired, right? So what has been feeling more tired than ever before? But it just felt different, Eric. And noticed some lumps and bumps in my neck, which turned out to be enlarged lymph nodes. I noticed fluid pooling around my ankles, which just seemed so unusual. This horrible abdominal pain, and it got worse and worse over the course of a couple of weeks. And I actually I was on an OBGYN rotation. I went from taking my medical school exam for OBGYN and walking down the hall to the emergency department. And so I don't know what's going on. And so they ran blood work, and I'll never forget, my doctor walked in the room and said, "David, your liver, your kidneys, your bone marrow, your heart, and your lungs are shutting down. We have to hospitalize you right away." And within days, I was in the ICU, had my last rites read to me because my doctors didn't think I would survive, and was on dialysis, and it just sort of got even worse from there.
Well, I know over the course, I think there was at least 5 times that you were courting death.
That's right.
Only a little ways away from that. And you were diagnosed with a form of Castleman's disease, which there's 2 forms, as I understand. You had the most serious multi-centric one. Maybe you could explain, since you are now the world authority, Castleman's disease. What is the difference between the more benign and the more, very serious one?
So sure. So the thing that's in common between both forms of Castleman's is they both look the same under the microscope. So if you cut out a lymph node, they look the same under the microscope. But the form that I have, what's called idiopathic multicentric Castleman disease, this is a deadly form where our immune system attacks and shuts down our vital organs for an unknown cause. And really the only way until recently that you could treat it is with chemotherapy. Versus the more benign form, though it looks the same under the microscope, behaves completely differently. Patients typically are cured with surgery. And it just sort of goes to show, I love following your podcast and the work you do, the importance of molecular medicine, right? Something might look the same under the microscope, but molecularly it behaves very differently, and therefore patients have completely different outcomes.
Well, and your presentation told us right from the get-go that you have the benign form, or the one that is so easily cured. We're going to get into cures because this is, of course, a theme of yours, and you went from chasing my cure to chasing cures for all.
Right.
One of the most memorable quotes about you was in the New York Times profile, where this is a few years back. An Emory University professor said, "I almost wish that every disease had a David to be part of the charge." And that says a lot right there. And it basically, in a nutshell, is kind of where we're headed with this discussion. So you have discovered something that helped you, having tried, I don't know how many different interventions, but you finally came upon something that really made a difference. You've had remission for about 8 years now. Is that right?
So over 9 years, that's right, on this drug that you know very well and that I know many have been talking about a lot, sirolimus, also called rapamycin, drug that was never intended for Castleman's and never been used before for Castleman's. But yeah, based on work that I did in the lab, I believe, and based on the data in front of me, that the mTOR signaling pathway was turned into overdrive, and sirolimus is a potent inhibitor of mTOR. And So I became the first patient ever with my disease to be treated with this drug. And yeah, over 9 years, and we've got patients all over the world that are now on it.
Now, how did you know to give rapamycin a shot?
So after my 4th relapse of the disease, I'd gone back to Penn Medical School, got another year of med school under my belt, relapsed again. That relapse was so difficult because I was on an experimental drug that blocks interleukin-6 and it didn't work. And so that's when I realized that if I wanted any chance of survival, that I would need to get involved in conducting research. And I knew it was unlikely I would find anything that could be helpful for myself, but I knew it was the only shot that I had. And so I started collecting blood samples on myself, storing them in the freezer every couple of weeks. Any chance I got, to go into the lab while I was finishing up medical school to work on these samples. And I relapsed and nearly died for a 5th time while I was collecting these samples. And thankfully, I got a combination of 7 different chemotherapies. They saved my life. But when I got out of the hospital after those 7 chemos, I went in the lab and I began performing a series of experiments. So first I did serum proteomics, where I measured over 1,000 different molecules in my blood for multiple time points. I did flow cytometry to look at the status of the various immune cells in my body, how activated, how not activated they were. And from these multiple data sets, there was what's called an mTOR signature. Basically, the proteins that were really elevated in my blood were the proteins that are elevated when this particular communication line called mTOR is turned on. So it was a signature. It wasn't proof, but it was a signature. And then I went to a lymph node that I'd had resected during my last relapse, and I did a really simple stain. It cost about $17 to run this experiment. And that experiment showed in black and white that this particular communication line, mTOR, was turned into overdrive. And so I took the results. I drove down to Washington, D.C., and I took the results to a doctor of mine at NIH, and I showed it to him. I said, what do you think? I've got this deadly disease. I keep relapsing. There's no way I'm going to make it to my fiance and I's wedding day unless we try something. I'm out of options. And he said, sure, let's give rapamycin a try.
Yeah, that's pretty remarkable because rapamycin, as you know, as well as anyone, and has a multiplicity of effects.
Right.
One is, of course, it can help with mitochondrial dysfunction. And we'll talk about the prospects there, and things like long COVID. But also, it can suppress the immune system, where it is a standard drug for transplant to prevent rejection. So, coming upon this and then getting the right dose that you weren't necessarily knocking out your immune response capability. So did you have to explore what if there was a dose that worked, and how would you know? Did you keep doing all these tests on yourself to figure that out?
Yeah, it's such an important question because just as you said, rapamycin is one of those drugs where the dose really, really matters. I know Peter Attia has shared on your show before that actually at a low dose, it might actually boost the immune system in some ways, right, as opposed to suppress it. And so you really got to get that right dose. So in my case, my disease, so Castleman's, literally my immune system attacks my vital organs almost as though they're foreign organs. My immune system attacks my kidneys and my lungs and liver almost as though they should be rejected because they were a transplant. And so we thought, let's start out with an organ transplant rejection dose. Let's start out with the dose that we give to patients who get kidney transplants. And that dose was too high. I mean, my immune system was completely wiped out. And so we started dialing it back until we got to a level where my symptoms were controlled. It's now been 9 years without a relapse, and I'm going to knock on some wood on my desk. Symptoms were controlled, but also that my white blood cell counts weren't just zero, right? We wanted to find that middle ground. And so it was a little bit more trial and error to get that right dose, and a little bit less, quantitative proteomics, but I think we found the right dose.
Well, and then also you took all the lessons that you had to form a Castleman's global network. So tell us about how you went from the N of 1 to the N of thousands.
Sure. So as you know, my book is called Chasing My Cure, but I think that was the wrong term. It should have been Chasing Our Cure, because it actually really wasn't me working on my own. Yes, I was the one doing the experiments in the lab, but I also had this amazing network of people around me from all over the world, just as you mentioned, a scientific advisory board representing nearly every continent in the world, physicians, researchers, patients, all working together with this common mission of, okay, we've got this disease that is deadly and relentless. Can we build a team that's also relentless to try to find solutions for it? And so we decided to do things a little bit differently. So rather than the typical approach of rare disease research where you raise money first and then you invite researchers to apply for it, we said, what if we do things completely differently? What if we start out by just asking physicians, researchers, and patients, what are the important studies to do? Let's come up with that prioritized list, and then let's go out and recruit the best researchers in the world to do those studies, whether or not they'd studied Castleman's or knew anything about Castleman's in the first place. Just, what should be done? Let's go recruit someone to do it. And that approach has really been groundbreaking, and it's been amazing to get patients participating in what research questions we're asking, to get researchers who had never even heard of Castleman disease, but they're the best people in the world at single-cell RNA sequencing working on our project. And it's been just this great example where when you bring the right people together and you have a really clear mission and vision, you can achieve so much more than I ever could have on my own.
Now, around the world, of the people who have the multicentric form of Castleman's, are they mostly taking rapamycin now?
So there's a lot of patients that are on rapamycin doing very well. There's also an IL-6 blocker, which has an incredible backstory that's also used quite a bit for Castleman's. I had been given that IL-6 blocker. It didn't work for me, but it does work for between 1/3 and 1/2 of patients. And so there are a lot of patients that are doing well on IL-6 blockade. And I have to quickly just share the story about IL-6. Many listeners will have heard about tocilizumab or IL-6 blockade in the context of COVID. It, as you know, is the first drug that is administered to patients in the intensive care unit after they've gotten dexamethasone. But the story behind tocilizumab is an incredible one, and it started with Castleman disease. Back in the 90s, Kazuo Yoshizaki discovered that interleukin-6 was important in Castleman's, and he developed this drug now called tocilizumab to block interleukin-6, the receptor for it. And I had heard that Kazu gave it to himself as the first patient ever. And I said, Kazu, I heard you gave it to yourself. He said, no, I didn't give it to myself. The nurse gave it to me. I said, oh my gosh, Kazu. So he discovered the target, he developed the drug, and he gave it to himself as the first human ever, which I don't recommend by any means. But he didn't, have a bad outcome, and he tested the drug in Castleman's. He got approval in Japan for Castleman disease. And then when it was purchased by a large pharmaceutical company, they decided Castleman's was too small of a market in the US. So they actually studied it in rheumatoid arthritis and juvenile idiopathic arthritis, now in COVID, cellular immunotherapy. So this drug, tocilizumab, that was made for Castleman's is now being used for myriad other conditions all over the world.
Right, yeah. And we recently were discussing about the prospects, because long COVID has been shown to have mitochondrial dysfunction, and obviously an immune that's really an untoward activation. And so the rapamycin may turn out to be a worthy candidate for long COVID. And, we really need to do a trial for that, basically repurposing what you found helped you and many others for something that now millions of people are suffering. Do you think there's a chance it will work there?
I think so. I think that just as you said, there's multiple mechanisms through which rapamycin may be effective. As we said earlier, the dose is really going to matter, which is tough when you do a large clinical trial because, you got to pick the right dose and have sufficient numbers on the right dose to be able to see the right effect and think about duration. We talked about that previously as well. Is it 1 month, 6 months, 1 year? What's the right duration to really see the effect that you're looking for? So I think there's a really good mechanistic reasons to believe that it could be useful. I think some of the observational data around metformin and metformin use during acute COVID and how that lowers the risk of long COVID also is circumstantial but supportive evidence, given that metformin works in many ways similar to rapamycin. So I think there's reason to believe. And as you can imagine, as someone who's alive thanks to a drug that wasn't made for my disease, I like to think a lot about the drugs that are at our neighborhood pharmacy that maybe were intended for one thing, but what are the other things that they could be helpful for?
Yeah, well, living proof of repurposing, if there ever was one. Now, that wasn't enough that you accomplished in your young age in your career. Now you've launched Every Cure.
Right.
So tell us about this, because this is something I know you made a major announcement at the Global Clinton Initiative recently. And this is taking lessons you've learned and all the people that you've had a chance to work with and taking to yet another mega level.
Oh, thanks so much, Eric. Yeah. So as you know, I'm alive thanks to a repurposed drug. And after finding that drug, I joined the faculty here at the University of Pennsylvania to continue to do that. So basically to continue to study hyperinflammatory diseases like Castleman's, and then to look for existing drugs to see if they could potentially be repurposed to treat those diseases right now, not 10 years from now, not $1 billion from now, drugs that are on the pharmacy shelf. And we've been really successful here at Penn. Our center has identified and or advanced 16 repurposed drugs for diseases that they were not initially intended for. And in the course of doing that, it's just with every one of these drugs and with every patient who walks into our center gets a drug that wasn't intended for their disease and then weeks later is doing better, it just continues to be, in my face that, oh my gosh, there are drugs sitting on the pharmacy shelf that could save patients' lives. And the reverse of it is that there are patients suffering when there's a drug sitting on the pharmacy shelf that could have saved their lives. And so the more that I've seen this and also, frankly, the longer that I've lived on this repurposed drug, the more compelled I felt to say, we need to do something systematically. We can't just do it, Castleman disease and then Poem syndrome, one rare disease at a time. We really need to do this at scale. And a few things kind of came together at once that led to every cure. So one piece of it is something that you've been tracking so well, and that is the incredible progress with generative AI and the incredible advent of these new tools that can help us to do things that took a medical student like myself months working in the lab. You can learn in seconds, frankly, utilizing artificial intelligence. And so that's one big pillar of this. So, well, I guess one pillar is that certainly we've had a lot of experience and success with repurposing. Another is that AI was really coming to the forefront and developing. And the third is that I got this phone call on March 31st of 2021, so the day before April Fool's Day, and I'm telling you, all I could think about is repurposing. And I get this call, and it's from an unknown number, and the person answers or is on the other line and says, President Clinton is on the other line. He read your book. He wants to talk to you. And it's the day before April Fool's Day, so I was pretty sure it was a joke. And then on the other line, I hear, David, it's Bill Clinton. And I'm like, oh my gosh, this really is, President Clinton. And so, he had read my book, and his question for me was, how many more drugs do you think are out there that could be repurposed? And I told him that I believe that, there's 3,000 approved drugs that are approved on average between 1 to 3 diseases each. But it's not an exaggeration to think that each one of those drugs could have maybe 1 to 3 additional diseases. And really think about that. That's doubling the impact of the pharmaceutical products within reach. So he said, well, what's your dream? And I said, my dream is to create an initiative to do this systematically, to unlock the full potential of every drug for every disease possible. And he said, well, if you ever do that, know that you've got my full support, and I'd love for you to announce it at the Clinton Global Initiative. And so he kept calling every few months to check in to see if I was going to do this. And eventually I did feel like we could build the right team to do this at scale. And so we launched Every Cure. We announced at the Clinton Global Initiative. And the last year has all been about building the team, bringing together the resources and the funds to create an AI engine that comes up with a predictive score for the likelihood of every drug to treat every disease. So all 3,000 drugs against all 20,000 human diseases. And this is the perfect use case for something like artificial intelligence, right? There's so much data that's already out there to train it on. You're looking for patterns in the data that can connect one drug to a disease. So basically, we come up with these predictive scores, and then we take the most promising ones into clinical trials to prove, does this drug actually help patients with this disease, and move it all the way forward, whether or not it's profitable for a company.
Yeah, I think this is kind of the thematic of the Medicine and Machine podcast, where how can we learn from AI and machines what can make for better medicine, better health? So one example of that, which I know you're familiar with, is during the pandemic, the JAK kinase inhibitor baricitinib was my data mined through AI to say, oh, this drug is being used for rheumatoid arthritis and alopecia areata, but let's try this in COVID, severe COVID. And it has, of course, in more than one randomized trial shown to save lives. It has full approval, and it is an AI success story in the midst of the pandemic. A lot of people think about vaccines and maybe Paxlovid, but this is, as well as saving lives. And of course, there are many others like this. And you mentioned the 3,000 drugs; those are approved drugs. There's another big jump of under 10,000 in the Reframe database here at Scripps Research. That these molecules, they didn't make it as a drug, but they were tested, and so that it could even be beyond the ones that are approved that didn't get approved that could turn out to be, great success stories. So you're onto something that's extraordinary, but it isn't just this that you're doing. I mean, because you get to work with Carl June and
Right.
the people that are doing all this T-cell engineering that are changing the world. So that's the drug side, but you're also working on cellular engineering. So can you tell us about that?
Sure. So I've had the opportunity to be first off at this great institution, University of Pennsylvania, where there is so much great work being done in cellular immunotherapy and engineering. My focus has been on how do you make sure that when you give someone cellular immunotherapy and they do have a cytokine storm reaction to it, how do we control the cytokine storm so that we can save their life, so that the cellular immunotherapy can extend their life? And so I've worked, thankfully, very closely with Carl and John Wary and others to think about sort of the tail end of cellular immunotherapy. And because, of course, if you can't control the cytokine storm when you give someone cellular immunotherapy and they die from the treatment, that's a travesty, right? So let's get them these life-saving therapies, but let's make sure that in the process of treating them with life-saving therapy that you don't cause more harm than good. And so we've been really focused on deeply profiling the cytokine storm that occurs with these cellular immunotherapies.
So this, I don't know if people are aware, the whole idea of CAR T, that is engineering T cells, is going through really a revolution, even now using CRISPR genome editing,
Right.
basically antigens that are specific to an individual's cancer, and revving up the cancer immune response beyond the drugs that we have today, like, the PD-1 receptor inhibitor. So, as you said, one of the biggest downsides is a cytokine release syndrome, which can be fatal. Is that now under check? Has that been markedly reduced over time?
It has been. It's been reduced tremendously thanks to IL-6 blockade, that drug tocilizumab I was telling you about earlier that Kazu made. It's pretty much given uniformly to patients all over the world. There are still some challenges. Individuals getting something called ICANS, which is neurological and quite progressive and serious. And so there's still unmet need in this sort of controlling the immune response in cellular immunotherapy, but it's a lot better than it was. As you said, the more powerful and the better we're getting with cellular immunotherapies, the more we have to continue to retool how do we prevent it from getting out of control. As you know, solid tumors, so when people think about typical cancers like lung cancer or colon cancer, those have been more refractory to cellular immunotherapy over the years. Blood cancers have been or been much more responsive to cellular immunotherapy. Now that we're starting to crack the code in solid tumors, I think that it may be a different sort of an immune response that we're seeing afterwards. And so we're sort of having to pay attention because, one, you need to treat the cancer, but two, you need to make sure they don't cause harm along the way.
Right. Yeah. So no question, the solid cancers are largely still, I mean, there's some small successes, but
You're right.
we got a ways to go. But one of the things that's noteworthy, you've got this every cure going for all these rare diseases.
Right.
And on the other end, you're also working on cancer, essentially, by trying to make the treatments that the group at Penn is kind of mission control here for this.
That's right.
Making that safer, more effective, hopefully eventually charting a path that's going to be effective in addressing cancers. This, of course, this whole theory about the immune system, which you have, again, knowing yourself, know thyself so well, as a source of why people get cancer and don't respond. For years that was out there. And finally, we started to have some checkpoint immunotherapy agents, which, as you've already gotten to, helps some people with certain specific types of cancer. But there seems to be so much more room to rev up our immune system. And so I wanted to ask you about the fact, is cancer, obviously it's a genomic disease, but is the ability for it to go on to metastasis, is that because our immune system is defective or just can't meet up to the challenge?
I think it's one part, maybe it's defective. I think it's another part that maybe it's not that it's defective, but that we can program it to be more effective, right? So it's not so much that it was, it didn't send something, but it's that there's actually human potential to reprogram these cells, just as we talked about earlier, to actually effectively remove them. I think that the immune system, of course, I'm very biased. I am an immunologist. I run an immunology lab, but I think that immunology and the immune system plays such a critical role all across medicine. I mean, look at the colchicine approval in cardiovascular disease. I mean, this is just such an incredible concept that, 20 or 30 years ago, would we have ever thought that colchicine, a gout drug that is certainly anti-inflammatory, would get an approval for secondary prevention of heart disease. And it's because of the role that inflammation plays in cardiovascular disease. I mean, I would love to hear your thoughts on colchicine in particular, given your background.
Yeah, I think it's a really interesting point you're making, which is here is another repurposed drug. I never would have thought I'd give colchicine for heart disease, but there's 2 randomized placebo-controlled trials that provide compelling evidence of benefit, whether it's post-MI or people at high risk. And I don't even think it's out there in the cardiovascular community. And the reason is because, as you've really centered on, is because people, when it's repurposed, they just don't get it. That there's these drugs that will help people. And you wouldn't exactly call ischemic heart disease or coronary atherosclerosis a rare disease, right?
No.
But the number of people I see in clinic who have coronary disease or post-MI who are getting colchicine is next to nil. So, it's going to take a while to hopefully convince people that those 2 trials actually do provide some compelling evidence. I think that cardiologists just think of it as, inducing diarrhea for treating gout or something, so and again, dose is another part of that story.
Yes.
Because there's a company that's trying to market it, which is different than generic colchicine.
Yes.
I think, as I recall, the difference between 0.5 and 0.6, which I can't believe.
That's correct. That's correct.
And so obviously that marketing it's kind of helped to cloud the issue, because it's not like it's a new drug. It's just a slightly different dose of a very old drug. Mm-hmm. Well, so where do you go now? I mean, you're starting to have impact globally. Are you going to move outside to the galaxy? What's the next step, David?
Oh, Eric, no, I so appreciate the kind words. I'm just so thankful to be here. I mean, just literally to be on your podcast with you. What an honor for me. But frankly, just to be alive. And I feel like I have this incredible responsibility that's like, I'm alive because of a drug that no one paid attention to, that was just sitting there all these years. And now there are patients all over the world that are alive because of other drugs that were just sitting there on the pharmacy shelf. So I feel this incredible drive and mission, which is to say, okay, every drug that's on the pharmacy shelf, we need to do everything we can to make sure that we can unlock the full potential of it, because, no patient should suffer if there's a treatment right there. And you brought up such a great point with colchicine. A lot of it comes down to incentives. And so, if the drug is cheap and old and generic, then no one's doing the work to figure out a new use for it. And so I feel so compelled for us to fix this problem. I think that the potential benefit to society is so great, to take drugs that are sitting on the pharmacy shelf. They're safe in many cases. They're inexpensive in most cases because they're generic, and to find new uses for them, because we know all too well that there are so many diseases that still cause so much hardship in this world. I mean, you mentioned long COVID, and there's obviously so many others that though modern medicine has solved so many problems, and we have a lot to be proud of. And I think AI is going to continue to help us to make even more of a dent. There's still a lot of work to be done. And so I feel this just incredible honor that I get to, spend every day working with scientists and colleagues to try to figure out, how can we save lives with cures within reach. And then the last thing I'll mention is what's so exciting about this approach is the turnaround time. So if I were to develop a new drug from scratch, I would need at least 10 years and somewhere in the order of 100 million, maybe closer to $1 billion to do that one drug for, first disease. We can repurpose drugs and discover a new use for a drug, and within weeks have a patient on that drug. And now, of course, the risk-benefit profile has to make sense. And I don't think it's a good idea to ever do data-free repurposing. It should always be data-driven repurposing and in the right context for the right patient. But the ability to reach and help someone within weeks of a discovery and to have drugs now approved for new uses and recommended after just a couple years versus, what would take so much more money and so much more time, it just sort of makes everything feel so attainable. And I think a lot about hope, because there have been a lot of times in my journey where I was out of hope, and I leaned on my family and my friends, and they gave me hope for the future and a dream for the future. But when I think about repurposing, at the end of the day, it's really about saying, there are all these drugs. Let's create some hope here by actually tapping into these drugs. And it may not be that there's a drug for your condition or someone that you love, but if you look on the pharmacy shelf, trust me, there are a lot of patients that can benefit from those conditions. We just have to look for them.
Yeah, so that's another question, is that because there's thousands, tens, thousands of conditions out there. And by the way, collectively, people with rare diseases like Castleman account for tens of millions of people
Yes.
around the world. How do you prioritize? I mean, obviously, you have a bent towards immunologic conditions or basis for conditions, but how would you say, okay, every cure is going for this one or that one? This drug looks so alluring. We got to. How do you make the call?
It's a great question, and it's a tough one. So we ran our first version of the algorithm, and we got 36 million scores. So every drug versus every disease. And so, of course, you look at the top and you say, wow, this makes sense. This drug works for this disease. You start going down. And to your point, how do you start picking out of 36 million? Obviously, you don't want to pick the ones at the bottom because, it's very unlikely they're going to work. But how do you pick out of the top ones? And so we think about it in terms of a number of criteria, but really I boil it down to two. The first is the likelihood of scientific success. So how clear is the mechanism? So you talk about long COVID and, how does the drug work? And maybe even if there's some clinical data that already exists. So, what's the scientific evidence? But then the second question is, what's the impact? And so what's so cool about being a nonprofit organization through Every Cure and being in this position, being armed with all the data, is that we don't have to worry about if the drug is going to be profitable in the new disease area. We don't have to worry about what's the cost of it. It's really about what's the impact. So you can think about impact in terms of the impact for a given life. So is it going to be potentially transformative for a small number of patients, or is it going to be, a smaller impact across a larger population? What is the cost of the drug to the healthcare system? You can start to factor in things around impact that drug companies, frankly, aren't able to think about because it's not part of their business model. And so, one, is it likely to help? And two, what's the impact that's going to come from this? And then that's where you have to build a great team and bring together a lot of great scientists. And I'll make a shameless plug that right now we're recruiting the best drug developers, the best data scientists in the world to be a part of our Every Cure team, because we know that we've got this great opportunity, but we need a great team to go through and to make these, to your point, what are going to be really tough decisions. Are we going to go after this disease, or are we going to go after that disease?
Yeah, no, I can see where it's almost going to be an unlimited opportunities that you're creating by having this systematic approach. And then, you want to have points on the board that every cure is actually curing some conditions that previously were not really approachable or had inadequate types of intervention. Well, this is, really just remarkable what you're onto now. I mean, I think this idea, which many people have heard about, how AI is the hottest area in life science industry for drug discovery. And there's 100 companies now with all partnerships with all the major pharma. Each major pharma has like 3 AI partners. But the difference, what you're doing at Every Cure is, it seems like, they're not so much interested in repurposing drugs. They're interested in coming up with new molecules that they can make a billion or many billions.
That's right.
Is that a fair comparison, or do some of those AI companies, are they also competing with Every Cure?
You're exactly right. We're using almost identical technologies, but those companies are purely looking for new molecular entities or tweaks to existing molecules, so that way there can be intellectual property. And so what's so exciting is that, so if you do that approach and you have a new molecule you're going after and it looks really promising, get ready to spend $1 billion on drug development and get ready to wait 10 years. And so we say, well, let's use the exact same technology, but let's point it at the drugs that are already at the neighborhood CVS, where we can do a clinical trial for $1 million, for one 1,000th the cost, and we can get it to a patient tomorrow. it's just, for me, it's like, why is there this gap? It seems so obvious. The technology is the same, but if you apply it to new molecular entities versus old ones, you have this incredible opportunity for immediate impact. So yes, your comparison is exactly right. And there's amazingly always been this gap in our drug development system. And it's something that I've spoken a lot with Janet Woodcock and others at the FDA about. And that's this, thing that's a bit surprising. And that's that once a drug gets a first approval, surprisingly, that's when R&D starts slowing down for that drug. You'd think that, okay, we've got an approved drug, it's on the market. Let's ramp up R&D. Let's find all the additional uses for it. But sort of paradoxically, now that the drug got its first approval, let's start ramping down R&D and let's, go find another drug. But in my opinion, as someone who's alive thanks to one of these old drugs, I think that's when we have to start ramping up and really focusing on finding more uses. And that's what we're trying to do with every cure.
Well, I don't know that anyone could be more of a force or a icon. I mean, you look like the picture of health. No one would ever believe that you were on death's door 5 times and went through all that you did from 2010 until 9 years ago. So it's really just so extraordinary. I think we're all indebted to you, David, you and the team that you're developing at Every Cure to help accelerate the knowledge base for taking these medications that could really help cure people that up till now have not been assessed or identified and proven. So this is really, it's just amazing. And you've been able to get some great funding from key players out there. So hats off to you. Big time kudos. We're going to be following your story because it wasn't over when you wrote the book. 4 years ago, chasing my cure, and now you're chasing everybody's cures. David, thank you so much for all that you're doing. The team at Penn with John Wary, Carl June, I mean, these are some real heroes, and to have you on as part of that is just, it's just, how does it get better? So I know you're going to add some more to the group, and watch out, folks. There's a lot, fasten your seatbelts here, because a lot of good things are going to come out of this. So thanks for joining today.
Eric, thanks so much for having me, and thanks for those kind words. It's truly such an honor to be with you, to be a part of this podcast. And like I said, I'm so thankful to be here, and I'm so thankful I get to have the opportunity to find drugs so that more people can be here and to experience all the amazing things in life.
You are listening to Medicine and the Machine with Medscape Editor-in-Chief Eric Topol and Master Storyteller and Clinician Abraham Verghese as they talk with experts around the globe about the hottest topics in healthcare. This podcast is intended for U.S. healthcare professionals only.